Very warm welcome to one and all. I am Amrita, Assistant Professor in Biochemistry from Sri Adi Sunjanagiri Women's College, Khammam. Today, I am going to present a topic on DNA fingerprinting. Let's start with definition. DNA fingerprinting is a hybridization technique which involves the analysis of nitrogenous base sequence in the DNA. It is used to identify the similarities in DNA molecules of two individuals. It is also called DNA colony hybridization or DNA profiling or molecular fingerprinting. Let's see the discovery of DNA fingerprinting. This technique was invented by Alex Jeffrey in 1985 in Leicester University, UK. Principle involved in this technique is in our human genome about 20% of genes code for protein. Remaining 80% are called junk DNA, that is non-coding regions of DNA which do not code for any protein sequence. It is further classified into two types, highly repetitive nucleotides and random nucleotides. It is a technique of determining nucleotide sequence of certain areas of DNA which are unique to each individual. Each person has a unique DNA fingerprinting. It relies on repetitive sequences that are highly variable called variable number of tandem repeats, particularly short tandem repeats. Families of variable number of tandem repeats. It is classified into two families. The first one is microsatellites, also known as short tandem repeats. They are repetitive sequence of 2 to 6 base pairs of DNA, and the other one is mini satellites. Mini satellites are also known as variable number of tandem repeats. Section of DNA that will consist of 10 to 60 base pair. Their analysis is useful in genetics, biology and DNA fingerprinting. This diagram represents the tandem repeat elements such as microsatellites and mini satellites. Steps involved in DNA fingerprinting are isolation of DNA, digestion of DNA, electrophoresis, southern blotting, probe labeling and autoradiography. Let's see the steps one by one in detail. The first step involved in this process is isolation of DNA. DNA of an organism which has to be tested is isolated. It is called host DNA. DNA must be recovered from cells or tissues such as skin cells, hair, blood and saliva. Second step is digestion of DNA. The isolated host DNA is treated with restriction enzymes. It cuts DNA into small fragments. This diagram shows EcoR1, which is a restriction enzyme of Escherichia coli, which cuts DNA at specific site and form sticky ends. The double stranded DNA fragments are denatured to produce single stranded DNA by alkali treatment. The alkali used in this process is sodium hydroxide. Electrophoresis. The DNA segments are separated by agarose gel electrophoresis based on their size. The DNA fragments are injected into the wells and electric current is applied along the gel. DNA is negatively charged, so it is attracted to the positive end of the gel. The shorter fragments move faster than longer fragments. As a result, DNA is separated based on their size. Southern blot. The electrophoresis DNA is transferred to the nitrocellulose filter paper by placing the sheet on the gel and soaking them overnight by the process called southern blot. Probe labeling. The nitrocellulose filter paper is then baked at 80 degrees Celsius for 2 hours. A known sequence of single-stranded DNA is prepared. It is called probe DNA. The probe DNA is labeled with radioactive isotopes such as phosphorus-32. Hybridization. The probe DNA is added to nitrocellulose filter paper containing the host DNA. The single-stranded probe DNA base pairs with complementary strands of the host DNA. As a result of hybridization, DNA-DNA hybrids are formed on the nitrocellulose filter paper. This diagram represents that adenine pairs with the complementary base thymine, guanine with cytosine. Autoradiography, the last step of this process. The nitrocellulose filter paper is photographed on the X-ray film by autoradiography. The film is then analyzed to determine the presence of hybrid nucleic acid. This diagram represents the overall process of DNA fingerprinting. DNA is extracted from the sample and restriction enzymes cuts the DNA fragments and it is electrophoresed. Then it is transferred to a membrane and incubated with probes, and X-rays are allowed to pass in the nitrocellulose filter paper. As a result, DNA fingerprint is formed. Applications of DNA fingerprinting. It has broad application in fields such as forensic science, parentage testing, medical diagnosis, food industry and agriculture. Medical diagnosis. Pathogens such as Neisseria gonorrhea, Vibrio cholerae and Staphylococcus aureus have been diagnosed by using DNA fingerprinting. In forensic science, it is used in detection of crime and legal disputes. In the crime scene, there are 3 suspects. DNA fingerprint of 3 suspects are taken and compared with the blood samples taken from the crime scene. Suspect 2 DNA profile matches with the blood samples taken from the crime scene. So, from this, it was concluded that suspect 2 is present in the crime scene. The remaining 2 suspects are get eliminated. In parenting disputes, it can identify the real genetic mother, father and the offspring. The mother, child and 2 father suspects DNA profile are being matched. 50% of mother and 50% of father 2 profile matches with the child. From this, father 2 was the father of the child. Hereditary diseases used to find genes connected with hereditary diseases such as hemophilia, Alzheimer's disease. Evolution helps in discovery of evolutionary distance exists between 2 adjacent groups in evolutionary tree. In sociology, it can identify the racial groups, their origin, historical migration and invasion. Thank you.